Assalamualaikum and hello everyone. We are still in chapter 9, Reproduction and Development. Today I'm going to explain about the next subtopic of the human reproductive system, which is fertilization and fetal development. In previous subtopic, you have learned about the male and female reproductive organ and how sperms and secondary oocytes are produced in the testes and ovary. Fertilization must occur before a zygote can be formed. The upper diagram shows fertilization process and the journey of zygote to become embryo. Secondary oocyte released from the ovary will enter fallopian tube. Fertilization, or also called conception in human, occurs when a sperm fuses with an egg or mature oocyte in an oviduct. Cleavage or cell division begins in the oviduct as the embryo is moved towards the uterus by peristalsis and the movement of cilia. When the embryo reaches the uterus, it will be implanted in the endometrium. At this stage, the embryo is in the form of blastocyst, which is a sphere of cells that surround a central cavity. The lower diagram shows the development of embryo from a single-celled zygote, which undergoes a series of cell divisions until it becomes a mature 38-week fetus and ready for childbirth. However, I'm sorry to inform you that most of you will fail in this subtopic. This is the overview of subtopic 9.3. It can be divided into two main phases, which are fertilization and fetal development. There are 4 stages in fertilization process, which are capacitation, acrosomal reaction, fusion of sperm head membrane and oocyte, and the last stage, cortical reaction. Zygote will develop into embryo and fetus in the process called embryogenesis. Embryonic development involves 3 stages, which are cleavage, gastrulation, and organogenesis. The learning outcome of this subtopic are, at the end of the lesson, you should be able to explain stages that lead to fertilization, define embryogenesis, state developmental stages from zygote to the formation of morula, blastocyst, and gastrula through cleavage, define organogenesis, and state the organ formed from each germ layers during organogenesis. What is fertilization? Fertilization is the fusion of sperm and an egg or mature oocyte to form zygote. It occurs in the oviduct, also known as fallopian tube. Sperms can live up to 72 hours in the uterus, while secondary oocyte will live up to 24 hours after ovulation. The diagram at the right shows the stages in fertilization, starting from the burrowing of sperm head into the secondary oocyte until the sperm's nucleus fused with the egg's nucleus. Fertilization occurs in 4 stages. It starts with capacitation, next is acrosomal reaction, and then fusion of sperm head membrane and oocyte, and the last one is cortical reaction. These 3 diagrams show the first stage of fertilization, which is capacitation and its analogy, and acrosomal reaction. Capacitation is the functional maturation of sperm through the female secretion in the genital tract. Certain molecules on the sperm are altered, for example, the removal of cholesterol coating from the membrane of sperm. It increases the motility of sperm and exposes the acrosomal cap for acrosomal reaction. In other words, capacitation is preparing the sperm to meet the egg, or its analogy, like the well-groomed or capacitated sperm that is going out for dating with an egg. The capacitated sperm is able to migrate through layers of granulosa cells or follicle cells, also known as corona radiata, and reaches zona pellucida. Acrosomal reaction is the digestion of zona pellucida by the enzymes discharged from the acrosomal vesicle via exocytosis. These neck diagrams show the process of fusion of sperm head membrane and oocyte membrane and the final stage of fertilization, the cortical reaction. When the sperm's head reaches the plasma membrane of the egg or oocyte, the specific membrane protein on sperm binds to the receptor on the egg cell membrane. The two membranes fused. The sperm nucleus enters cytoplasm of egg and triggers cortical reaction. In cortical reaction, lysosome near the secondary oocyte's membrane, known as cortical granules, release enzymes into zona pellucida via exocytosis. This enzyme will thicken and harden the zona pellucida to form fertilization envelope. Fertilization envelope will block other sperms from entering the oocyte to prevent polyspermy. The diagram shows stages of fertilization after capacitation, which are acrosomal reaction, fusion of sperm head membrane and oocyte, and cortical reaction. This diagram shows the overall process of fertilization, starting from capacitation until cortical reaction. Once the sperm nucleus enters the egg cytoplasm, the tail falls off. The entry of sperm stimulates secondary oocyte to complete meiosis II. Ovum and second polar body are formed. At this stage, each nucleus of the sperm and ovum is known as pronucleus. Fusion of male and female pronuclei produce a diploid zygote. DNA synthesis begins, and the first cell division occurs to produce multicellular zygote that will develop into multicellular fetus. As a conclusion, in sexual reproduction, meiosis halved the chromosome number in gametes, and fertilization doubled the number again in single-celled zygote. The zygote will undergo cell divisions to form multicellular fetus through embryogenesis. After fertilization, zygote undergoes embryonic development, or embryogenesis, involving 3 stages, which are cleavage, gastrulation, and organogenesis. Human life cycle begins with, after fertilization of gametes, the single-celled zygote will undergo embryogenesis, which consists of cleavage, gastrulation, and organogenesis. After birth, the baby will develop until he or she reaches maturity. Human will produce the next generation by sexual reproduction. Embryogenesis is the process of formation and development of embryo until birth. The process occurs during pregnancy or gestation. Duration of pregnancy in human is around 38 weeks, and it can be divided into 3 trimesters, each of about 3 months. Embryogenesis consists of 3 stages, which are cleavage, gastrulation, and organogenesis. Cleavage is the continuous rapid cell divisions of zygote in the oviduct during early embryonic development. It multiplies the number of the cells, but not its size, until it becomes a solid ball of cells called morula. It is still surrounded with zona pellucida. Each cell is called as blastomere. The cell continues dividing mitotically to produce a hollow ball of cells called blastocyst. Implantation of blastocyst in the endometrium occurs around 7 days after fertilization, when zona pellucida disintegrates. Blastocyst has a fluid-filled cavity called the blastocele. The blastomeres differentiate into outer layer, the trophoblast, and inner cell mass. Gastrulation is the formation of gastrula from blastocyst after implantation occurs. About 2 weeks after fertilization, the inner cell mass in blastocyst will be rearranged. It is characterized by migration of the cells inward to form a 3-germ-layered embryo. The inner cell mass will differentiate into ectoderm, mesoderm, and endoderm. This diagram shows the embryo embedded in the endometrium and consists of the 3 germ layers, ectoderm, mesoderm, and endoderm. Organogenesis is the process of organ formation in embryo. Organs arise from the 3 germ layers. Neurulation is the initial stage of organogenesis. It is a process of neural tube formation where it will develop into brain and spinal cord. The fate of the 3 germ layers means they will differentiate and develop into different organs. The ectoderm will develop into many different organs, for example, nervous system, skin, and epithelial lining of oral cavity and rectum. The mesoderm develops into muscle and skeletal system and many internal organs, whereas the endoderm develops into lining of digestive and respiratory tracts and lining of urinary bladder. The specialization of the 3 germ layers will end the development of fetus, and after 38 weeks in gestation, the fetus is matured and ready to be delivered. For further comprehension of the subtopic, you may refer to many international books, especially Campbell and Solomon. I will share with you an animation video on fertilization. I hope it will give you more apprehension on the subtopic. Now, let us test your knowledge on this subtopic with a few simple questions. Scan the QR code and answer the questions. Good luck!